Hello, and thank you for joining this OncLive TV peer exchange. This program will feature expert panel perspectives on unmet and emerging needs in the treatment of lung cancer. My name is Corey Langer. I'm the Director of Thoracic Oncology at the Abramson Cancer Center and Professor of Medicine at the University of Pennsylvania. Participating today on our panel are Ross Camage, Associate Professor of Medicine, Oncology, Director of the Thoracic Oncology Clinical Program, and faculty member of the Developmental Therapeutics Program, as well as the Associate Director for Clinical Research at the University of Colorado in Aurora. Ramaswamy Govardhan, Co-Director of the Section of Medical Oncology and Professor of Medicine, Division of Oncology at WashU in St. Louis. Roy Herbst, Ensign Professor of Medicine, Professor of Pharmacology, Chief of MedOnc, Director of the Thoracic Oncology Research Program, and Associate Director for Translational Research at the Yale Comprehensive Cancer Center in New Haven. And Alice Shaw, Associate Professor, Department of Medicine at Harvard, attending physician in the Thoracic Cancer Program at MGH. Mark Socinski, Director of the Lung Cancer Section, Division of Hem-Onc, and co-director of the UPMC Lung Cancer Center of Excellence at the University of Pittsburgh. Thank you again for joining us today, and let's get started. The first section that I want to cover is the evolving role of cytotoxic therapy in advanced non-small cell. We've seen the recent approval of Nab-Paclitaxel, a nanoparticle albumin-bound taxane, in the frontline setting. So let's begin by briefly discussing the potential role of this agent in frontline treatment. Mark, if you could start.
Sure. Obviously, we know there are several taxanes that have activity in non-small cell lung cancer. Part of the problem with these agents is that they're water insoluble, so they have to be dissolved in solvents. These solvents, in the case of paclitaxel, really involve chemophore and alcohol. And there are some obviously drawbacks from that. The nanoparticle technology in binding this active agent to albumin, I think, is a physiologic advantage. I kind of personally view bulky tumor sites as albumin sponges, and we've understood kind of the transport aspects of this. We don't understand much about the biomarker issues of these albumin pathway-related sorts of proteins, but the Nab-Paclitaxel trial was really designed to go head-to-head against solvent-based paclitaxel combined with carboplatin. In the phase 3 trial, the control arm was solvent-based paclitaxel and carbo, given in standard doses and standard frequency. Obviously, that regimen requires standard pre-medications for hypersensitivity reactions. The investigational arm was a Nab-Paclitaxel weekly administration with the same dose and schedule of carboplatin as the control arm. No standard pre-medications were given, and the Nab-Paclitaxel was given over 15 minutes or so. So there's a convenience aspect to that, lack of pre-medication as well as the infusion time. Because paclitaxel was already approved as an agent in non-small cell, the FDA allowed overall response rate to be the primary endpoint. And this trial met its primary endpoint. The difference was a response rate of 25% on the control arm of solvent-based paclitaxel versus 33% on the investigational arm. So that led to it being a positive trial. Now, we did choose to try to get some sense of PFS and OS as secondary endpoints, but there was not a significant difference in that setting. Since we live in the world of histology, we looked at response rate based upon histology, and in the non-squamous population, there was really no difference in response rates between the two arms. If you look at the other three populations that we defined, which was squamous, large cell, and then the other category, there was a significant difference in favor of Nab-Paclitaxel versus solvent-based paclitaxel. And then in squamous, the difference was between a 24% response rate on the solvent-based arm versus a 41%. And this was all independent radiologic review. The biologic reasons as to why you should see this dramatic difference in squamous versus no difference in non-squamous are poorly understood. But I think that really kind of gives it a clinical niche in where you would think about using that drug in that population where very few options exist.
What about the elderly?
The elderly was another observation that was actually quite striking. Of the 1,050 patients or so, about 160 or so of them were over the age of 70. And there was a dramatic benefit in overall survival, almost a doubling of the median from about 10 months for carbosolvent paclitaxel versus almost 20 months for the Nab-Paclitaxel arm. Again, the biologic understanding of that is a little hard. Is it just, is there something there, or is it just, a spurious observation? It should be repeated. There isn't going to be an effort to look at specifically in the elderly, but not necessarily in a way to kind of sort this out. Is this a better regimen for older patients?
So really the optimal trial, if you were to proceed, would be to compare a weekly regimen of solvent-based paclitaxel to a weekly, the weekly approach with Nab-Paclitaxel in the elderly.
Yeah. The QUA regimen versus the the FDA-approved regimen right
And
now.
that'll at least sort out, is it the schedule or is it the drug, or are they absolutely equivalent? Ross, I know you have...